Recently, on the CFUW national website, I discovered that not long after the founding of CFUW, when Madame Curie visited the US in 1921, CFUW made a substantial contribution to a fund raised by women to purchase radium for her research, and also funded her trip to Niagara Falls. With our speaker tonight being in the medical research field, I thought this anecdote was of particular interest. Next year, on March 16th, our own CFUW KW Club will celebrate its 100th birthday. Over the decades, our club has continued to fulfill our founder's legacy, to develop camaraderie through interest groups, advocate on important issues, as I mentioned earlier, the homeless, FSDA, the environment, to name a few, and also the crisis in long-term care, and to encourage girls and women to pursue a higher education. A charitable fund was established to provide scholarships and bursaries, and this fund has grown to just under $1.6 million, with over $40,000 awarded through 49 scholarships annually to university, college, and high school students. Now back to our history. Following the founding of CFUW, British and North American university graduate women, having witnessed the devastation caused by the First World War, understood that even in safe countries like Canada, when strife and conflict spread in other parts of the globe, security can dissolve without warning. Today, we witness the turmoil caused by the current global pandemic and how it has affected millions of women, not just here, but tragically in the underdeveloped world, where women already marginalized economically have lost their source of income, are preventing from entering schools, and suffer human rights violations. Those visionary women conceived the idea of a worldwide organization of university women, and in 1920, their dream became a reality with the International Federation of the University Women, IFUW, now GWI. Today, thousands of women graduates throughout the world share the same principles that were inspired by those visionary leaders and by the founders of CFUW. Over the decades, CFUW, KW, CFUW, and GWI have worked tirelessly to encourage women to pursue higher education that would enable them to take their place in leadership positions in the private sector and in government. Tonight, let us reflect on three questions. 100 years ago, could we have imagined that the vision of the founders of CFUW and the numerous advocacy efforts of women and the men that supported them would position women in senior decision-making roles in the private sector, schools, universities, local and world government? Could our founders have imagined the extent of their success? Could we have imagined that we would witness the appointment of women as leaders in positions of power and influence on the global scene? Positions that could, in time, lay the foundation for a more humane and peaceful world. Can you imagine? That was quite something, wasn't it? So all your hands up. A very special thank you to a few people that worked to share that with us. First of all, to you, who sent the international section of that video for us to share. And then to Barbara Spronk and Sarah Gritzfeld, who worked together to seamlessly take the music and create that whole local video for us. Barbara, I'm so grateful for what you did. And to Sarah, you did an amazing job. That was very, very professional. And I'd like to all of us to give her an applause for doing that. So thank you. The next general meeting will be on March the 16th, as mentioned earlier. And I'd like to conclude the business for this evening. So may I have a motion to adjourn? Peggy Rose, seconder. Carol Smith. The meeting is now adjourned. I would like to now call upon Chris Cameron to introduce our speaker for this evening.
That was quite wonderful. We have one more wonderful woman to add to that group. Tonight, it's my pleasure to introduce Dr. DeWitte-Orr. Stephanie DeWitte-Orr is an associate professor in the departments of health sciences and biology at Wilfrid Laurier University. Her research focuses on innate antiviral immune responses, studying how cells detect when they are infected with viruses and how they defend themselves from virus infection. Stephanie received her PhD from the University of Waterloo, and then she did a postdoctoral fellowship at McMaster University. Dr. DeWitte-Orr is a recipient of numerous awards. She is the recipient of the Laurier Early Career Research Award, and I hope I'm saying this right, the Robert G. Boutier New Investigators Award from the Canadian Society of Zoology, and the Young Scientist Award from the Society for In Vitro Biology, in which she was the first Canadian recipient. Dr. Stephanie DeWitte-Orr has published numerous academic articles. I read through them. There was lots. Besides her role as an educator, she is presently leading a team in the development of an antiviral drug that will be a game changer in the treatment of all viruses. She studies these responses in numerous animal models. Thus, her work has impacted environmental, agricultural, and human health applications. So now I'm going to turn it over to Dr. DeWitte-Orr for her talk. Thank you very much.
Thank you very much. Can you all hear me okay? Lovely.
Yes.
Okay. Before I begin, can I just, I know you're running a little late. Do I need to have a hard finish of 9 p.m.?
I don't think so.
Or
No.
can we go a little over just in case?
We can go a little, yes.
Just so I
can.
know.
Yeah.
Okay, just so I know what to expect. Should I be rushing through some slides or not? Oh, we did that. Okay. All right. Thank you so much for having me this evening. It's wonderful to celebrate women and education. That's really close to my heart. So I'm very happy to hear like-minded women. And I'm very happy to talk to you this evening about coronaviruses. So Christine asked me to speak to you about the coronavirus and about vaccines. And I'm going to talk a little bit about antiviral drugs just because that's what I do. And I'm going to try to do it in a reasonable amount of time with a reasonable amount of depth. So if there's something that you just want some clarity on, please feel free at the end to ask me questions and I can go back and clarify. So let's just start with the picture on the slide right now. This is SARS-CoV-2. This is the virus that's causing all the trouble right now. This is an electron microscope picture. So I'm sure you might have seen pictures of the coronaviruses before. They have these spike proteins, these large proteins that stick out from the surface. So each one of these is a separate virus. And SARS-CoV-2 is, of course, the virus that's causing COVID-19, which is the what's making so many people sick around the globe. So what is a virus? Let's start there. Viruses are really, really small. I can't overstate that. They're a small particle made up of protein and nucleic acids. Nucleic acids are either DNA or RNA. And sometimes they have some fats in there as well. And that whole, so they have a protein and fat shell, and inside they have some sort of nucleic acid, either RNA or DNA. And they're so small that you can't see them under a normal microscope. You can only see them under an electron microscope. Actually, until 1930, we didn't even know what a virus looked like. We have been looking at bacteria since the 1600s, but we didn't even know what a virus was actually a virus until the 1930s. So a virus is this small entity that enters our cells. It needs our cells to make more copies of themselves. And so it enters our cells, it uses the cell to make more machinery. And just so you know, a cell is the building block of our bodies. Our body is made up of cells. And so the viruses are going to get into those cells, they're going to replicate, and then they're likely going to kill the cell when they leave, when they've made enough copies of themselves. Now, both the virus and the immune response to the virus is what could make you sick. So it's not just the virus that can make you sick, it's actually sometimes the immune response. And with this virus in particular, it's the immune response that's making people sick when the virus gets into the lungs and starts causing a strong immune response in your lungs. And that's what's causing you to be sick. So what does this virus look like? So I said viruses are really, really small. And SARS-CoV-2 is 120 nanometers in diameter. Now, 1 nanometer is a billionth of a meter. And a human hair is 75,000 nanometers in diameter. So a human hair is 75,000 nanometers. And a virus, this virus is 120. So now you can see it's so small we can't even see them with a normal microscope. The virus is actually a pretty big virus with RNA for a genome. So inside it's got the protein shell and it has RNA on the inside, and this RNA virus actually codes for almost 28 different proteins, which is really a lot for an RNA virus. Some RNA viruses make 4 proteins. This one makes 28. The reason why I'm telling you that is because that amount of proteins that virus makes means that it can do a lot of things that some viruses can't do. It can manipulate the immune system, our immune systems. It can shut things down, it can turn things on. So it has a lot more control of what's happening in your body than some other kinds of RNA viruses. So I've showed you what SARS-CoV-2 looks like, but what is it? So it's a beta coronavirus. Now, you've all heard of coronaviruses by now, and I think it's interesting that we all know coronaviruses, because coronaviruses are actually not very well studied viruses. If you think about viruses, you'll think of HIV or influenza. Those are very well studied viruses. Coronaviruses are not. Some virology textbooks don't even have coronaviruses in their textbooks. So we don't have as much information about coronaviruses as we do about some other kinds of viruses. But we know SARS-CoV-2 is a beta coronavirus. We know it's about 88 to 90% similar to 2 bat coronaviruses. So that kind of gives us an idea of where this virus came from, that it came from a bat. It's about 80% similar to the SARS virus that emerged. That's the language we use for when a virus shows up in the human population, so starts infecting humans. So there was a SARS-CoV-1, we just called it SARS until now, that emerged in 2002. So the SARS-CoV-2 that's circulating right now is about 80% similar to the one that came out in 2002. Now that one was interesting because you'll notice it only infected 8,000 individuals, and it had quite a few deaths, 808 deaths. So it had a high mortality rate, but it didn't infect a lot of people. And the reason for that is that SARS infected your lower respiratory tract, so lower in your lungs. The SARS-CoV-2 that's around now infects your upper respiratory tract. So it's much easier to spread a virus if it's in your nose, because when you sneeze, it comes out a lot easier. So it was similar to the virus now is similar to SARS-CoV-1, but not exactly the same. And then there's another beta coronavirus that causes serious disease in humans. It's called MERS. You might have heard of it. And it came from Saudi Arabia. And so the virus that's around now, SARS-CoV-2, is only 50% similar. And now there's a word that we use for animals that can support a virus inside of them, but they don't get sick. And that's the word reservoir. And so we think that for SARS-CoV-2, the main reservoir was the bat. So that was the animal that kept this virus, and the virus stayed alive in the bat, but the bat didn't get sick. For something like MERS, interestingly enough, we think the reservoir was a dromedary camel. So a different animal kind of supported that virus to stay alive. So we think, I think my next, we think that SARS-CoV-2 came from bat or bat guano, bat poop, with or without an intermediate host. And I'm going to show you again in another slide what that means. So what does this virus do when it gets inside you? So here's our virus here at the top. You can recognize it because of the spike proteins that are sticking out. You can see it as the RNA on the inside. And you'll hear in the news a lot about spike proteins. And the spike proteins are important because, for one, they're on the surface of the virus. So if you're developing a drug, you can target the surface of the virus easily. The second reason why the spike is important is because it's what binds to the cell. It's what helps the virus get in. And remember, viruses need to get into your cells in order to make more of themselves. So this virus binds to the ACE2 receptor on the surface of your airway cells. So when you breathe in the virus, its spike proteins are going to bind to the surface of your airway cells. And that's how it's like a lock and key. When it binds to the ACE2 receptor, it's like the door opens and the virus is allowed into the cell. And once the virus gets in, then it starts making more copies of itself. It copies its genome, its RNA, and it makes copies of its proteins. And then it assembles into the virus particles, and then those particles get released. So that is the replication cycles, what we call it, of the SARS-CoV-2 virus. Now, SARS-CoV-2 is better at binding to the ACE2 receptor than SARS-CoV-1, which is one of the reasons why it's more transmissible, or you can catch it easier, because it can get into cells better, it can replicate better, and it can make more of itself then. So I said that this virus came from a bat. How does that work? You might hear in the news something called if virus jumps. The virus jumps to a new, it jumps into humans. Well, obviously, you can see pictures of these viruses. They don't have legs. They can't really jump. So what does that mean? It means, we use a word called zoonosis. That's when a virus mutates in such a way so that it can infect a new host. So host is in this case, an animal that supports the virus to be able to infect. And if the reservoir is a bat, how did it all of a sudden infect a human? Well, it mutated. And viruses mutate all the time. That's what they want to do. They want to mutate to, maybe when they mutate, they'll gain a new function. They'll gain some new skill that they didn't have before. So they definitely try to make mistakes when they replicate in order to mutate. And what might have happened is that in December of 2019, there was a coronavirus in a bat, and that coronavirus mutated when it was copying itself. And then the new mutated virus now has gotten a new capability. Its S protein now can bind and enter human cells. Now, at the same time that this mutation was happening in a bat, there had to be a human who got in close contact with the bat and inhaled maybe some bat droppings. And then that new virus got into the lungs of the human, And now, because this virus has this new capability, it can infect the human and start replicating. So zoonosis is the word we use for that ability to change to a new host. And you might have heard that there might have been a bat source, a pangolin, which is an awesome animal. If you haven't looked them up before, I highly suggest looking at them. They're amazing animals. There's also a little bit of news about a snake. I think we've refuted the role of snakes in zoonosis, but this is a brand new article that just came out this month. And it just shows you how complex the possibilities are for, where did this virus come from? But it is likely that there was a virus that came from a bat, the coronavirus. It may have had, so the reservoir was a bat. There may have been a pangolin involved as an intermediate host. There may have also been another mammal as an intermediate host. And then somehow we got this coronavirus that now could infect humans. So it's a complicated story. We don't have the answers, but it's likely a bat, either directly or through some sort of intermediate host. Okay, but of course, that's not the only variant or not the only mutation that's happened with this virus. Viruses can mutate a lot. It's very normal. Influenza, HIV, those viruses mutate with incredible frequency. HIV is the most variation, the virus with the most variation on the planet that we know of in humans. It has incredible mutation capabilities. Coronaviruses actually don't have those same capabilities because they have so many proteins that they code for. Their genome is quite large. They actually have an enzyme, it's called a proofreading enzyme, that fixes mistakes when the genome is copied so that you don't get a lot of mutations. So we'd actually be probably in a lot more trouble if this virus didn't have this proofreading enzyme. So, but viruses mutate all the time. And many of these mutations are going to result in daughter viruses. So you have the original virus, and then you have the daughter viruses that are produced when the virus replicates. Those daughter viruses may have no advantage, or they may be worse at infecting, replicating, or transmitting in their host. But some of the mutations that happen are going to give the daughter viruses an even better advantage. And so that is why viruses mutate, for the chance of there being an advantageous mutation. And so I don't know if you know, but the first variant of the SARS-CoV-2 virus actually emerged last January and February. So we're hearing a lot about variants right now, but in fact, the first variant came in January, February of 2020. It was called D614G, and it actually completely replaced the original form of the virus by June 2020 across the globe. So the variant that's circulating now is actually something new, different from the Wuhan variant. And the D614G is actually had more infectivity and transmission than the original strain of the virus. So this is very normal. It happens with viruses. They're going to mutate randomly, but it's actually a sort of adaptation to the new host. So the virus is just going to kind of get used to being in a human, and it's going to make mutations along the way. Now, there are some variants that have made the news. Obviously, cluster 5 emerged in Denmark in mink farms in September of 2020. And this was more of a problem for minks than it was for humans. So that one is not so much of a worry for us. But there have been 3 variants that I'm sure you've noticed that have been in the news: B117, which emerged in the UK, B1351, which emerged in South Africa, and P1, which emerged in Brazil. Now, these are important for 2 reasons. One reason is because these mutations are all in the S protein, so then that spike protein, the part of the virus that helps get into the cell. So if you get mutations in the S protein, it could be that the virus can't infect the cell at all, or it could be that the virus can infect the cell better. And so for these 3 variants, it looks like they might actually be better at infecting, or at least for the B117 and the P1 strains. So they look like they're even better at infecting humans. With B1351, that doesn't seem to be the case, but the problem with that B1351, I'm trying to avoid using geographic locations, because when you talk about viruses, you don't want to talk about where they came from, because it gives negative connotations to the places where it arose. And really, you have very little control about where a virus arises from. So for the B1351 strain, it may not have changed transmissibility, but it is affecting how well the vaccine works with that strain of the virus. So mutations not only can affect how viruses infect cells, but they can also affect how well vaccines work. Because if the new variant is very different from the vaccine, then you're not going to get protection. So, okay, so now we have these viruses. They have this virus, has infected humans. It has gained the capability of infected humans. It's adapting to humans. How does it transmit? How do you catch this virus? Well, it's a respiratory virus. Okay, so it infects your airways. And particularly in what's makes SARS-CoV-2 so easier to catch is that, like I mentioned, it's in the upper respiratory tract. So it infects your upper airways, which means that you're going to get lots of virus in the top part of your airway. It's much easier than to sneeze or cough and transmit it to somebody else. So that's why you could catch SARS-CoV-2 so much easier than SARS-CoV-1, for example. The other thing that's another characteristic of SARS-CoV-2 transmission is that you have 5 to 6 days between when you get infected and when you have symptoms. Usually it's 5 to 6 days. It can be up to 14 days. That window of 5 to 6 days is a problem, because that's the time where you are infected, but you don't know. And unfortunately, you are infectious. You can give it to somebody else in that window. So even though you don't know you're sick, you could be getting other people sick. Now, on top of that is the problem that there are some people who get infected and actually never know they're sick. And those are called asymptomatic carriers. So these are people who are sick, can give it to somebody else, but don't have any symptoms. And they think up to 44% of all transmission could be due to asymptomatic carriers. We believe that transmission is primarily close contact, so you have to be close to somebody, and the virus is being transmitted by respiratory droplets. So that means the virus is in fluid that comes out of your nose and mouth, and it suspends in the air, but the droplets are quite not large, not gross large, but they're bigger than an aerosol. So aerosols are tiny little droplets that are in the air that can be suspended for long periods of time. And it's not quite clear yet whether this virus transmits by aerosols, but it definitely transmits by respiratory droplets. Another thing that I don't have on this slide, but I think is important, is that what's unique about this virus is that 80% of new cases come from 20% of existing cases. What I mean by that is most people who get this virus don't transmit it to anybody else. It's just a few people who transmit it to a lot of new people. And so you've heard of super spreaders, and that is a unique characteristic of this virus. And we don't really understand how that works, but it is something really different about this virus. Now, I just want to take a little side note here and say that masks do work. So if you're thinking, okay, this is air in the air, how do I protect myself? Masks work. And it's kind of funny, because when I was first interviewed in the news about masks, when they first started to come up, cloth masks, And I said on the news, you know, don't bother with cloth masks. They're not going to do anything. But in fact, they do something. They do help trap those larger respiratory droplets. They trap them in the mask so that they don't transmit easily to someone else. They actually also may act as a barrier the other way around to protect the wearer from air particles in the air. Now, the most effective cloth masks have multiple layers of tightly woven fabric. This is a balance. I have some masks that I'm wearing and I can barely breathe through them. They're so tightly woven. So, you know, you have to find a balance there, but it has to be something that, you know, you can't see through, with multiple layers. And fit is important. Please cover your nose and mouth with the mask, not just your mouth, because it's not effective if it doesn't cover your nose as well, because your nose is in your airways. And I wanted to show you this one study because I thought this was really fascinating. This was a study that looked at different states in the United States, and they compared COVID-19 cases and deaths in states that required masks and in states that did not require masks. And what they found was there are 10 times more cases in the states that didn't require masks than those that did. So and the flip side of that was, if you, in the states that wore a mask, there was 90% less cases. Additionally, deaths were reduced by 80% in the states that required mask wearing. So masks work, even if they're uncomfortable and they fog up your glasses. Okay, so now we have this virus. It has adapted to get into your airways. I've showed you how it replicates. I've told you how it transmits. What if it gets inside you? What happens? How are you going to feel ill? And I'm sure everybody's aware that SARS is the severe acute respiratory syndrome. It affects your airways primarily, or at least first. So your clinical manifestations are going to be fever and cough. And then there's labored breathing, muscle pain, headache, and then interestingly, diarrhea, which is a secondary site of infection for this virus. So it can start in your airways and it can move to your gastrointestinal tract. Now, what kind of treatments do we have? Unfortunately, not very many. So if you do get this virus, you're going to be given respiratory support if you need it. So put on a ventilator. And one of the best treatments we have at this point is an antibody transfusion. So essentially, patients who have gotten better from this virus will donate their blood. The antibodies will be taken from that blood and given to other patients who are really sick, and the antibodies will help fight the virus. So there's really not a whole lot of treatments that are available for the virus. This is the Johns Hopkins University COVID-19 dashboard. This has been following the pandemic since the first days of it emerging from Wuhan. And so you can log on here anytime, it's free, and you can see what the status is globally, you know, what country's winning or losing for the infections. And you can see as of a few days ago that we're at almost 108. I'm sure by now we are 108 million cases and 2.3 million deaths. So, you know, this is a serious pandemic that we're dealing with right now. How do we protect ourselves from this on a population level? How do we protect our population? So we protect it by herd immunity. Herd immunity is when a certain proportion of the population has immunity. Immunity means they won't get sick from the virus. And when you have, so let's look at two herds, two different populations. In this herd, a lot of individuals here are healthy, that's the light blue, but they don't have immunity. There's a few individuals who are immune to this virus. So that means they've either been vaccinated or they've had the virus. Their immune response has responded and they have memory. And so when people come into a herd or a population like this, where many people are not protected or not immune, you can see how quickly the virus can spread, because it's very easy for these contagious people to interact with healthy people without immunity. Now, if you have herd immunity, then you can see how many people here are immune to this virus. They're not going to get sick if they interact with the virus. There are a few people who are not immune, and they're going to be protected by all of the people who are already immune. So when this person who's infected comes into this population, it's not a high chance that they're going to run into someone who's actually doesn't have immunity. So this herd immunity protects those in the population who can't get vaccinated. And so this is the basis for vaccinations, is to not only to protect the individual, but also to protect the population, as long as enough people get vaccinated. So for SARS-CoV-2, we think we need at least 70% of the population to be vaccinated in order to get herd immunity, to protect those who can't be vaccinated. Now, the other strategy of herd immunity is just letting the virus go, just letting it infect the population. And Sweden tried that. And that did not work very well for them, because this virus is not a virus we know very well. It can cause devastating effects in different populations, such as the aged population. And so a vaccine is the much safer approach to get to that 70% protection in order to protect the whole, our population as a whole. So what is a vaccine? How do we get this protection? So a vaccine is either, when you're thinking about viruses anyways, a vaccine is either a live virus that's been weakened. It's a live virus. It's a live virus. It's an inactivated virus or an attenuated virus, something that's been weakened, or pieces of a virus, either the protein or that RNA or DNA. And it's used to stimulate the immune system. So what the vaccine is doing is it's trying to tell your immune system that this is a virus you need to pay attention to. This is a virus that you need to create memory for. So when a vaccine is first put into your arm or up your nose, or however the vaccine is administered, your innate immune response is going to detect it first. And your innate immune response is going to decide whether to respond to the vaccine or not, and how to respond. Now, once we've passed the innate immune response, it's going to activate the adaptive immune response. And the adaptive immune response includes T cells and B cells. You might be familiar with those. T cells are used to kill virus-infected cells, and B cells make antibodies to kill the virus. And so you need both of these to be activated to get a really good immune response. Now, it's the adaptive immune response that has memory, and that's what we really want to harness with the vaccine, is memory. You want your immune system to remember this virus, so that the time you actually see the virus for real, you're protected. So what is immune memory? So after your first, if you think about something like chickenpox, when I was a kid, I was taken down the street and introduced to the girl down the street with chickenpox so that I would get chickenpox, so that I would never get chickenpox again. That's memory. After your first exposure to a virus, your T cells and B cells, your innate immune system gets activated, then your T cells and B cells get activated, then most of them die at the end of the infection. But 5 to 10% of your T cells and B cells stay alive. And these are the cells that remember that virus. So when you're reinfected with that same virus, those memory T cells and B cells will activate faster, stronger, and better, so that you wouldn't even know you're sick. The B cells and T cells will activate and get rid of that virus immediately. So your vaccine that you got was that first exposure to the virus. It was a safe exposure, much safer than getting the real virus. And it's going to establish memory so that the next time when you're going to the grocery store or you're talking to a friend and you get sneezed on and that virus gets into your airways, you have memory B cells and T cells that activate right away and get rid of the virus. So that's why vaccines work. Now, an effective vaccine is one that establishes long-term memory, so memory that lasts. And unfortunately, not all vaccines provide long-term memory. For example, influenza vaccines, they don't provide long-term memory. They only last for about 9 months, which is why you need a new vaccine every year. But if you get a great vaccine, you can have lifelong protection against that virus. So the real key here is that when you get infected with an actual virus, your memory B cells and T cells activate quickly to protect you so that you don't even get sick. Now, I wanted to just spend a moment here to talk to you about the difference between a vaccine and an antiviral drug. So a vaccine is something that you give a healthy person, so they're not infected with the virus, to help establish immune memory to protect you from future possible exposures to that virus. So you can get an influenza vaccine or this coronavirus vaccine. You're healthy, but your immune system is going to respond and is going to remember that virus. So if you actually see the real virus, you're protected. Now, an antiviral drug doesn't establish memory. It doesn't help your immune system remember that virus for another exposure. An antiviral drug is something that you either give before an infection, so prophylactically, or after you're infected, that's a therapeutic drug, to stop the virus. The drug may target the virus itself, or it may activate the immune response. Antiviral drugs do not induce memory, so it's not like a vaccine. Some examples of antiviral drugs you may have seen are Tamiflu is a very common one, acyclovir, ribavirin. Unfortunately, there are not a lot of antiviral drugs available because the virus, when it infects our cells, it takes on the cell as its own. So trying to create a drug that kills the virus but not the host cell is actually really difficult. So there's not a lot of drugs available. So vaccines are definitely our forefront, strongest strategy to control the current pandemic. What's in a vaccine? Well, it depends. It depends on the vaccine. This is a really great infographic from the British Society of Immunology. Most what's in a vaccine is water. After that, you're going to get the active ingredients, so pieces of the virus. And I'm going to talk to you a little bit about the coronavirus vaccines that are being available now, what's being developed, and what kind of active ingredients are being developed. But there's going to be pieces of the virus, and that's what your immune system is going to recognize. Now, that's not the only thing inside a vaccine. You also need something called an adjuvant. Now, some vaccines are self-adjuvanting, which means the active ingredient is the adjuvant as well. But remember I talked about that innate immune response that decides whether to pay attention to the vaccine? The adjuvant is what makes the innate immune system pay attention to this vaccine. If you didn't have that adjuvant, your vaccine would do nothing. It would just break down in your body and you wouldn't get an immune response. So you need the adjuvant to wake up the immune system and pay attention. There's also some preservatives and stabilizers. This is to keep vaccines safe. You see a lot of these more so in multiple dose vaccines as opposed to single dose vaccines. But there are some preservatives and stabilizers, and there also might be residual traces of things that were needed in the manufacturing. But all of those are safe below concerning levels. So that's what's in a vaccine. Do vaccines work? And I'm sure you can guess as a virologist, I definitely believe that vaccines work and that they're appropriate to control virus infections. These are just a few timelines you can see here of different kinds of pathogens. So diphtheria, pertussis, and measles. Measles is a particularly difficult virus that can infect people so easily. I told you that coronaviruses, the herd immunity is you need about 70%. For measles, you need like 95% of the population to be vaccinated in order to get herd immunity because of how transmissible that virus is. And you can see as long as everyone gets vaccinated, that virus disappears from the population. So vaccines work. The gold standard story, of course, is smallpox. Smallpox killed 500 million people in the last 100 years of active infection. It killed more people than both world wars. It was devastating, but it's eradicated. That means it's not replicating or transmitting between people on our planet anymore because of vaccines. And polio is another one. Polio is almost completely gone from our planet because of the polio vaccine. So vaccines, of course, do work. For the current virus, SARS-CoV-2, ever since the virus first emerged and started to look like it was going to be a big problem, there were immediate responses to develop vaccines. And there's been a lot of different strategies to create a vaccine. This is a really great paper from Nature that just looks at all the different kinds of strategies. I'm just going to go through them really quickly just so you understand the diversity of the kinds of vaccines that are being produced. The first vaccine strategy is called a live or inactivated virus. Now, we don't use a lot of live virus vaccines, but we do use a lot of inactivated or attenuated viruses. This vaccine strategy is our most common vaccine strategy, for sure. What we do is we grow up a lot of virus, we inactivate it or weaken it in some way, and then you put that into somebody. This is a really good vaccine strategy because you get the real virus, you get a real immune response, you usually get really good memory. The problem with this vaccine strategy is that you need a lot of virus, and coronaviruses are really difficult to grow in culture. So we actually can't pursue this vaccine strategy very well for SARS-CoV-2. Another problem is that this kind of vaccine strategy actually causes a lot of immune response in the lungs. It's called eosinophilia of the lungs, and it's a really bad side effect. So there are vaccines using this platform in China and India, but the US and Canada have not approved these strategies for human use, at least not yet, which is unfortunate because this is our known strategy. This is the strategy that we use for vaccines, and we can't use it for this virus. So that's a problem. The next strategy, using the real virus and weakening it, okay, we can't use that. The next strategy is, what if we take a virus that we know is safe and we genetically engineer it so that it puts spike proteins on its surface? Okay, so it's a real virus, so you'll get a good immune response, but it's safer because it's not coronavirus. In a lot of cases, it's called an adenovirus. So it's a safe virus that has been engineered to make coronavirus spike proteins on its surface. So it's likely going to be safer. The problem with this vaccine strategy is that you can really only use this vaccine once per person. After they've mounted an immune response to this vaccine, the next time you try to use it, it's going to remember that vaccine platform. It's going to remember the adenovirus and it's going to shut it down. So this is not a vaccine that you can use a few years later on the same person. Another problem with this vaccine strategy is there's a lot of proteins in a virus that's not the spike protein. And so you get something called a dilution effect. So you don't get as strong of an immune response as you could with some of the other strategies. This strategy of using viral vectors or using something like adenovirus as a carrier has been used for gene therapy, but it's never been used before now for human use for vaccines. This is the platform that AstraZeneca is using for their vaccine. So this is the vaccine. that the UK has developed. It's being used in the UK and is anytime going to be approved for use in Canada. Now, if we simplify vaccines even further, well, remember what a virus is made of. It's made of protein and nucleic acids. So what can we make a vaccine out of? Protein or nucleic acids. So the protein is either pieces of proteins. So this is a protein subunit vaccine. So you can take just the spike proteins or the M protein from the coronavirus, and you can put it into as a vaccine and see if you get an immune response against that. These protein subunits are they're easy to make. Well, easier to make. They're very safe. They don't usually induce a strong immune response. Now, Novavax is using this protein subunit vaccine platform. So this is the Novavax vaccine. This is the one that's going to be hopefully made in Montreal. So this will be our first Canadian-made vaccine. The way Novavax's works is they actually don't have the proteins kind of floating around like this. They have the proteins sort of sitting in a fat bubble, sort of like a virus-like particle, but not quite. And so it's a little bit different than what this looks like, but it's kind of the same principle using the spike protein as a vaccine. So then there's these virus-like particles. This is actually a really cool technology where you take a virus, you take out the nucleic acid, and we just use the shell as the vaccine. It works really well. You get a good immune response. It's got a good safety profile. They're incredibly difficult to make. You can't make them fast enough. So we actually have virus-like particle vaccines for hepatitis B and human papillomavirus, but we have none for coronaviruses right now. We don't know how to make them for coronaviruses. So that's unfortunate because that would have been a pretty nice platform. And finally, there are the nucleic acid vaccines. So viruses are made of proteins and DNA or RNA. And because coronaviruses are RNA viruses, and also because RNA is actually a little bit easier to get protein made from than DNA, the two nucleic acid-based vaccines that are currently available in Canada from Moderna and Pfizer, they're RNA vaccines. So in that case, we have a piece of RNA and, oops, sorry, I did that, piece of RNA, and it is covered in a fat bubble. And that fat bubble is going to help the RNA get into the cell. It gets into the cytoplasm of the cell and it gets translated into proteins. Those viral proteins are the spike proteins, and they're what's going to stimulate the immune response. Okay, so these vaccines are relatively easy to make. I doubt Moderna and Pfizer would say they're easy to make, but relatively. They can, the nice thing about these RNA vaccines is they can rather quickly accommodate variation. So remember we talked about some of those variants, particularly the one from South Africa, that looks like it's not going to be a very effective, especially the AstraZeneca vaccine may not be good at establishing memory against that variant. But with a vaccine like this, where it's just RNA, you can change that sequence rather quickly and make a new sequence that matches the new variants. It's pretty safe. There's no virus here. It's just RNA. You need that fat coat, that lipid coat, to get it into the cell. And unfortunately, these complexes are not very stable. So you usually need to keep them at very cold temperatures. And that makes it difficult. Not as difficult in developed countries like Canada and the US and Europe, but much more difficult in developing countries. Again, this is a platform that's never been approved for use in humans. So we're brand new territory here. And this is the platform for Pfizer and Moderna's vaccines. So I just wanted to stress how amazing it is that we have vaccines this quickly for coronaviruses, because we've never made a vaccine for coronaviruses before. Influenza, we're good at. We're great at making influenza. Well, we're pretty good at making influenza vaccines. We've got all the infrastructure in place. We can have tons of embryonated eggs. We can make vaccines really quickly. So when H1N1 in 2009, when the swine flu emerged, We created a vaccine, but the virus emerged in April, and by December of that year, anyone who wanted a vaccine for that flu virus could get one, because we knew how to make that vaccine. We knew the platform. Unfortunately, we've never made a coronavirus vaccine before, which is why you can see we're trying all kinds of different platforms, all kinds of different strategies, because we've just never done it before. And there are some challenges with coronaviruses. Unfortunately, coronaviruses, when they infect humans, they don't usually give us lifelong immunity. So if you get infected with a human coronavirus, you usually have protection for 2 to 3 years. And then after that, the memory B cells and T cells die. You don't get memory anymore. So this is going to be a real challenge long term, is to be able to have a vaccine program that supports herd immunity over time. We still don't understand SARS-CoV-2 immune memory. It does seem like asymptomatic carriers don't have immunity. People who get very mild symptoms only have very short-lasting immunity. So this is going to have serious implications for keeping that herd immunity at 70%. None of the front-runner vaccines, none of the vaccines that are being used right now are on proven platforms. We've never used them before. They've never been proven in humans before. And we don't have proven safety data. Well, that's a little concerning. Although, I mean, I know that the FDA and Canada Health is working their best to keep these, of course, as safe as possible. But we are rushing vaccine development. Vaccine development usually takes 6 to 10 years, and we're trying to do it in a year. So it's moving quite quickly. And there's a lot of people working on vaccine development. This is from the New York Times. You can look at it yourself on a slow Friday night. The coronavirus vaccine tracker. And you can see there's a number of vaccines, lots and lots, that are in the different phases of testing. And these are global statistics. So there's 4 vaccines that are approved for full use around the globe, and 4 that have been abandoned because they're not safe or they're not effective. And of course, these 2 numbers are going to change depending on how these studies go over here. The 2 approved vaccines in Canada are the RNA vaccines from Moderna and Pfizer. They both have incredible efficacies of 95%, which is, I mean, that's just amazing for a vaccine. You need 2 doses. You get your first dose, and then you have to wait 3 to 4 weeks, and then you get your second dose. And they need to be stored at very cold temperatures because they're RNA and they're unstable if they're not stored at cold temperatures, which isn't so bad for Canada, not great for some other places in the world. Coming down the line is the AstraZeneca vaccine. That's that adenovirus, that virus vector vaccine where they've engineered a safe virus to make spike proteins on its surface. It's not as effective, but it's pretty good at 82% efficacy. You still need 2 doses. And the problem with 2 doses is that you need compliance. For one, you need that person to agree to come back, but also 2, you need to not run out of vaccine, right? Which is what we've done now. You can't give out vaccine and not save a second dose for those individuals. So 2 doses is proven to be problematic. The AstraZeneca vaccine can be stored at 4 degrees, which is much more reasonable. And then the Novavax, that's that protein vaccine. That's the vaccine that may be made in Montreal. It has about an 89% efficacy so far. It also needs 2 doses, and it can be stored at 4 degrees, so much more reasonable temperature. Okay, and I just wanted to finish. Oh, I'm not quite done yet. Okay, so I just wanted to finish to tell you a bit about the drug that we're working on. So we're working on a drug, we're not working on a vaccine. What we've done, I don't know if you remember, but I talked about innate immunity and adaptive. Adaptive has memory. Innate is what decides what to respond to. And what we did was we've put an innate immune stimulant on a nanoparticle. And this triggers really strongly, it strongly triggers the innate immune response. And if you can activate the innate immune response, you can have a broad spectrum protection. What that means is innate immunity is not specific to one virus. B cells and T cells are, but innate immunity isn't. So if you activate the innate immune response, you're going to protect not just against coronaviruses, but against any virus. So we envision this to be something you inhale maybe once a week or once a day to protect yourself. Your airway cells will be activated, and then the virus won't be able to infect. This is what we're envisioning anyways. So I wanted to show you some pictures here. These are human lung cells that we grow in the lab. And I may be biased, but I think they're absolutely beautiful. With those beautiful, healthy cells, you can see they're all individual cells. They've got nice membranes. So this is your lung cells healthy. These are your lung cells on coronavirus. They're not there. They died. They're all dead because the virus got into those cells, replicated, and then escaped by killing the cell. Now, these are those same cells, but with the drug. So this is human lung cells with the virus, but with the drug, and they're completely protected. So we think we've got something pretty exciting here. Now, this is actually 14 days post-infection. That means that we gave the drug, then we added the virus, and even 2 weeks after, we still saw no virus present. So we're seeing good protection in human lung cells. Okay, and my final slide here, I just wanted to reiterate what you can do to protect yourself against this virus. Remember, it's an airway virus, so cover your face with a mask, wear something that fits well to your face. That's going to hold the virus in to protect others and hopefully protect you from the virus if it's suspended in the air. Stay 2 meters apart. We know definitely from studies that the virus is much more easily transmitted when you're close to somebody. Okay, so stay at least 2 meters apart. Sanitize and wash your hands frequently and get vaccinated when you can. And that's the end of my presentation. Thank you very much for listening.
Dr. DeWitte-Orr, thank you very much for your presentation this evening. Does anybody have any questions? And would you prefer that they put their hands up or maybe a question in the chat line?
It doesn't matter.
Okay, so if I'll allow you to pick off the screen then, if anyone's got their hands up. Let me see if I can change my view to gallery view.
Oh, I have a question.
Okay.
Will this be something that do you foresee us having to take a vaccine every year, just like the flu vaccine? Yeah.
Yeah, we don't know yet, but it does seem as though the vaccine is, immunity is not that long lasting. And it's quite unlikely that a vaccine is going to provide better protection than someone who's actually got infected. Like, you're going to get better immunity from, or better memory from, an actual infection. So it's quite likely. I'd be surprised if the vaccine would provide more than a year or two of protection. So it is quite likely that we're going to have some sort of yearly, or every 2 years, some sort of boost.
I think Jenka has a hand up, and so does Dinah Cherry, and Herda Park has a comment.
Can I go? So I'm very interested in this notion of vaccine resistance. Speaking because I don't think we've got enough information about the coronavirus vaccines, but say for influenza vaccines, is there any evidence to suggest that having had the vaccine, even though it might not be 100% effective, that it actually blunts the effect of the illness should you get it? Should you get the virus? Does the vaccine actually moderate the effect of the illness?
You mean if you don't have a perfect match?
That's right. Yeah.
Yes. So there's definitely going to be some protection. It's just not going to be as good as a perfect match. And it's going to vary depending on the strains. So if you think of protection as maybe a circle on a graph, and then a variant, if it's overlapping a little bit, so it's kind of the same, you're going to get some protection. If it's way over here and very different, then you might not get any protection. So it depends on how different the variant is.
I guess, but does it actually affect, even if you have the vaccine, but you don't have a great, it's not 100% effective, will it affect the your body's response to respond to the virus and blunt the effect?
Okay. No, so your B cells and T cells, so that adaptive immune response that you want to get from a vaccine, they're incredibly specific. The memory that they establish is very specific to that strain of that virus.
Okay.
So it's not going to, if there's no match, it's not going to make you worse at responding, but it's not going to make you any better.
Okay. All right. Thanks. Thank you.
My question was, could you repeat the 80-20? You said 80% of the infected people will
come from 20% of, so 80% of new cases come from 20% of existing cases. Yeah, so that's really not normal for respiratory viruses. That means that out of 100 people of existing cases, 80% of the new cases just come from 20% of the existing. That means that 80% of the rest of us didn't transmit to anybody, or very few people. And we don't know why that is. Why are some people super spreaders and some people aren't? And I doubt it has a lot to do with hygiene. Like those are the real big sneezers, you know, or those the ones that don't cover their mouths. We don't exactly know why some people are really good at transmitting the virus and others aren't. But it's a unique feature of this virus.
And I have a question too. I have been advised that there is this product called betadine or betadine, and it comes both as a nasal spray and a gargle. And this comes from somebody who deals in, you know, these sorts of things, epidemiologist, infectious disease information. And she says that if you use this nasal spray and gargle before you go out, just, you know, several minutes before, it will coat the mucus linings of your nose, for example, and your throat, just to give you extra protection. Have you heard of this?
No, I haven't. No. And I think at this point, we're all trying to find things to protect us better. And I can't make any comments about that one in particular. Anything that lines the airway, of course, would be beneficial to a certain extent. I can tell you some viruses have really good strategies of breaking through the mucus layer, and coronaviruses are one of them. So they have an enzyme called neuraminidase that breaks down the mucus so that they can infect airway cells. And so it may or may not have a benefit. I can't say about that one.
Thank you.
You're welcome.
Stephanie, we have some questions in the chat line. For example, one is, how and when do you use your antiviral drug? Another one, what type of vaccine is the Johnson & Johnson? And then a few others. I can go
Okay. So, I can see them. I can answer these. Okay.
Thank
so how and when do you use our drug? I don't know yet. We're still developing it. We're going to be moving to preclinical trials soon. But the idea would be some sort of inhaler that you would inhale, and then that would activate your airway cells and protect them. It give you protection by mounting this innate immune response. But we still have to figure out dosage and timing and all that kind of thing. So I don't know yet. Okay, what type of vaccine is the J&J vaccine? I believe it's a virus vector vaccine. So the same thing as AstraZeneca, where it's a virus that's been a safe virus that's been engineered to make coronavirus spike proteins. Thank you, Christine, yeah. Will you be sharing your slides? I can. I can share them. I can send them to Christine if you'd like them.
Thank you.
Yeah. Thoughts about ACE inhibitors? Oh, for blood pressure regulation and severity of SARS-CoV-2. Okay, so you know, because ACE2 is not just a coronavirus surface receptor, it's also involved in blood pressure regulation. People who are on these drugs, there was a concern that there might be more of a susceptibility or maybe even a resistance against infection, and there doesn't seem to be a correlation between the two. So if you're on an ACE2 inhibitor, that doesn't seem to be one way or another to affect you as an individual and your susceptibility to the virus. The second question is, given that AstraZeneca vaccine has been approved in the UK, EU, Australia, Japan, India, Brazil, Japan, and Mexico, what do you think is the reason for the delays in the Health Canada approval? Well, I don't know, because I'm not in that field. But I think Canada is just trying to do its own due diligence. I think it's important to make sure that it's appropriate for Canada, that it's safe and that we feel comfortable having it here. So I think it's going to come any day, to be honest, that vaccine will be allowed to be administered in Canada. Okay, so you mentioned ACE2 receptors in your early slides. Is there any study into blocking the receptors? Yes, there are studies, and actually there are antibodies that are being developed that are as drugs to block the receptor. The problem is that receptor is not just a receptor for the virus, it's also important for just our body's functioning. So if you block the receptor, you're going to block important things that that receptor does for the body. So you have to be very careful about something like that. All right, can you get a negative reaction to the vaccine? Yes, you can. And so whenever you're considering a vaccine, you always want to weigh the pros and cons, the benefits and the risks. And so for something like the smallpox vaccine, for a long time, it made sense to get the vaccine because the risk was way higher than, the risk of the virus was way higher than the risk of the vaccine. But after the virus started, decreasing in the population, the vaccine was actually more risky. So then they stopped doing vaccinations. Because vaccines activate your immune system, they're not neutral. There's going to be side effects. So you have to decide what's riskier, being vaccinated or not being vaccinated and getting the infection. And yeah, there's always going to be, unfortunately, side effects with vaccines because of the nature of what they are. Why are patients in ICU treated prone in bed? Is that because the virus is an upper respiratory infection? I don't know the answer to that question. I'm assuming it's just
So weak.
No. Yeah, I think they just, they're laying down. Can the 2 doses of the vaccine be from 2 different manufacturers like Pfizer and Moderna? So it's possible that you could mix, have one from one vaccine, one from the other supplier. They're not exactly the same, and it's never been tested as a combo. So I don't think at this point we could mix and match. It's possible in the future if the companies ever decided to collaborate. It's a problem with medicine and business is you've got companies involved and they want to make money. And so, I don't know if anyone is ever going to approve that sort of a study in those companies. Are vaccines safe for people who are immunocompromised? No, not usually. So that's why we need herd immunity, because there's always going to be a subset of the population where vaccines are not safe. So you need to protect those people. And so if the people who are healthy, who are capable of being vaccinated, get vaccinated, then those of us who are not eligible will be protected because of herd immunity. Can someone who's allergic to seafood safely have these vaccines? Yes, you should be able to, but I would definitely check with your doctor before getting vaccinated. But that should be fine. Why do people get a more severe side effect with a second dose? That's a great question. So the reason why you get 2 doses with a vaccine is because the first dose wasn't good enough. It didn't activate the immune system enough. So that's why you need a second, you need a boost. So the first one kind of gets the ball rolling, but the second one is the one that really solidifies the immune response. And I would say maybe they don't have their dose exactly perfect and they overshoot. So their second dose is establishing memory maybe a little too well. So you get too strong of an immune response and you can get some of those side effects. So I think it's still safe, but that second dose is there to give that strong immune response, and it's doing what it's supposed to do. It's just those side effects are from your immune response. Those feelings of achiness and muscle soreness and fever and things like that are all parts of your immune system responding to the vaccine. So doing what it's supposed to do. Given that there are several vaccines that will be available, should we be concerned about which one to get? So when the RNA vaccines came out, as a scientist, I'm trained to be critical. That's my job, right? I'm supposed to be critical of technology, scientific technology. I'm supposed to find the holes so we can fill them and make it better. So when the RNA vaccines came out, well, the first one, when Pfizer came out, I was very skeptical because it's a brand new platform. We don't know a lot about it. When Moderna's phase 3 trials were completed and they were almost identical to Pfizer's, I felt much better. I felt like, okay, 2 independent trials from completely separate companies getting the same results. That looks really good to me. But I was still kind of wary. But now we're seeing the AstraZeneca and we're seeing the Novavax, all these different platforms. And in my mind, I'm like, okay, if I ever get a choice, which one do I want? I want the RNA vaccine, please. I want those Moderna and Pfizer vaccines. So I don't think we'll ever get a chance to choose. But if I did have the choice, I'd pick the RNA vaccine. I like it. It's simple. It's got a nice, great efficacy. That second dose is a bit of a doozy, but other than that, I think that that's a real nice vaccine. But of course, you know, they're not going to be approved if they're not safe and efficacious. So I think they're probably going to all be okay. And I don't think we're going to get a choice. It's my kids getting daycare. You get what you get and you don't get upset. I think that's it for questions in the chat.
Okay, Stephanie, thank you so much. I think we've been looking forward to this presentation for weeks. I don't even have to say why. I think a number of us saying that can we sort the wheat from the chaff? Can we really understand from a professional exactly what is going on? So you provided us with a very thorough presentation. So I'm certain that around having coffee tomorrow, I'll be trying to explain all this to my husband and not doing a very good job. But I would like to ask Peggy Rose to please say thank you to Stephanie.
You can hear me? Everybody can hear
Yes.
me? Good. Yes, thank you, Dr. DeWitte-Orr. This has been fabulous. I'm so pleased that you are able to be our speaker tonight. Not everybody knows that we had Dr. DeWitte-Orr has filled in for us. We usually give people quite a bit more notice, like months and months worth of notice. And Stephanie was able to fill in for us on very short notice. So we really appreciate that you were available and that you agreed to fill in for us tonight. I don't think we really imagined a couple of weeks ago that we would still be at this point, but also that your presentation tonight is so timely for addressing our concerns, our ongoing concerns about COVID-19. We hear about this every day, morning and night, from a lot of different people. So it's really refreshing and very nice to be able to get the facts from you, somebody that we know we can trust, and that you're able to honestly give us all this information. I was very interested in hearing about what's in a vaccine and the very simple breakdown of the vaccine. I think it gives me a little peace of mind for when I eventually do get the vaccine. It's mostly water, but I feel a lot better knowing having the information that you've given us, and all the time and the work and the strategy going into the development of the vaccine, and the pros and the cons and the research and the frustration. I know we're all concerned about the actual rollout and the delivery of the vaccine, and I think with the information you've given us tonight, it helps us be a lot more comfortable with what we will eventually get. So I'd really like to thank you for sharing your knowledge with us about this, what you call COVID-19, and I call wicked, horrible virus, and all about the development that's going on for this much awaited vaccine. So come on, Novavax or Novavax, come on. And also thank you for making time for us tonight.
Oh, it was my pleasure. Thank you very much for having me. I'll send the slides to Christine so that anyone who wants them can get them from her.
Thank you. That would be very much appreciated. Thank you, Stephanie. Well, ladies and gentlemen who are with us this evening, that really ends the meeting, but we will keep, there's a couple of thumbs going up. And again, just before we close, and then we can have a bit of social time, I want to thank Grace Hollett from National, Elena Freeman from CFUW Ontario, our RD, for joining us this evening. And again, a special thank you to Sarah for that incredible video. The messages are coming in on the chat line, and I'm sure that we'll continue to get some incredible feedback. Susan Gentile has already asked if we could post it on Facebook, so we need to talk about that. Roberta Bilak and Chris Bailey, I just want to acknowledge again that they're willing to step into the executive committee next year, and I will be chasing up a few others and see if you'll come and join us as well. Have I forgotten anybody? Hopefully not. So enjoy the rest of your evening. Thank you.
Thank you.
Thanks, Anita.
Thank you.
You have everybody on mute.